Hello, everyone. This is Lab 20. This is looking at the inheritance of the tendency to have high or low cholesterol levels. We're going to analyze different forms of the FH gene by DNA gel electrophoresis. It's not working. Recall that cholesterol can be a biological membrane component where it fits in between unsaturated fatty acids of glycerol phospholipids. It can also serve as a precursor for steroids, can be converted by bile in the liver in order to break down digested fats, and that's the only way cholesterol is excreted by bile. Here's a picture of cholesterol. Okay, unfortunately I could not do the slideshow. The computer wasn't responding, but this is a picture of cholesterol here, okay, where its rings will fit between unsaturated fatty acids within glycerol phospholipids or sphingolipids. As shown here, you have some unsaturated fatty acids attached to your glycerol phospholipids, and they don't want to be near each other. But with the cholesterol, it fits in between them and makes a tighter fit and makes the membrane a semi-solid. This is a picture of glycerol phospholipids. We have 4 different types here. In black is glycerol, CH2OH, CHOH, and CH2OH. But in position 3, or where this CH2O group is, there's a polar group. Okay, usually starts with a phosphate. And then here we have choline. Here we have ethanolamine, phosphatidylserine. And then we have another glycerol molecule here. And then in blue are your fatty acids, which remember there's an ester linkage between glycerol and the fatty acid. Then there'd be many CH2 groups and then end with the CH3 groups. Some of these fatty acids, as mentioned earlier, would be unsaturated. Cholesterol is also a precursor to steroid hormones and some vitamins, including vitamin D, which is very important that we have. It helps with bone development when you're young, and when you're older, prevents osteoporosis. It's a precursor to glucocorticoids, which helps with metabolism of carbohydrates, lipids, and proteins. It's also for aldosterone, excretion of water and salt from the kidneys, and helps and is responsible for looking at sexual characteristics or giving sexual characteristics such as testosterone. So it's important to reduce cholesterol in the bloodstream so we can reduce our chances of heart attack and stroke, as well as chances of Alzheimer's disease and reduce chances of kidney disease. There's a lot of work on your kidneys if you have a lot of cholesterol in the bloodstream. Okay. Sometimes the reason I have LDL here on the PowerPoint is that they used to say LDL was a bad cholesterol, but that's the protein that's carrying cholesterol to other tissues besides the liver, and therefore doesn't get excreted and clogs your arteries. FH, which is familial hypercholesteremia, is an inherited dominant disease which can lead to high levels of LDL, lipodensity protein, low density lipoprotein, mutation in the LDL receptors, so there's less or no LDL is taken up for the cells, and therefore you can't make steroids, hormones, or bile. So there are mutations in the LDL receptors that are in the membranes of cells, and normally those receptors would bring in the cholesterol when the cell needs it, but if there's a mutation there, they never bring the cholesterol in, and these people have abnormally high levels of cholesterol, even if they did not eat any food with cholesterol in it. This is a dominant disease. I don't know how many of you have had genetics recently, but it's a dominant disease where you only need one mutation in one of the 2 copies of the gene, to see problems with uptake of LDL containing cholesterol due to that defective receptor I mentioned, which again can lead to premature heart attacks, strokes, or kidney failure. Remember, you get 2 copies of each gene from each parent, 1 copy from each parent. If you only have 1 copy of the gene that dictates high cholesterol, then you're heterozygous, and then your other copy does not say high cholesterol. If both genes are mutated so that they both have a tendency to give you high cholesterol, it leads to early death in the 20s without treatment. That's if you're homozygous for both high cholesterol genes. Nowadays, they know to test for that and give you cholesterol lowering drugs. In this picture, we see the little blue molecules, or the LDL receptors on membranes. They bind LDL, which contains cholesterol inside of it from the bloodstream. The whole package comes in, and then as we can see here, the receptors can be recycled in times of need for cholesterol, or they could actually go to the lysosome and be degraded if the cell's full of cholesterol. But in any case, we see the cholesterol is released from these vesicles, or LDL, and then the cholesterol can be used to do work as part of the membrane components, precursor to various steroids, including vitamin D, as mentioned earlier. If you're homozygous for FH, that means you have both copies of the high cholesterol gene. Serum levels of cholesterol can be as high as 600 milligrams of cholesterol per 100 mils of serum. Normal levels are below 200. Here are some various cholesterol ranges from your lab manual, but these may be out of date. Here's showing you structure of DNA. Why am I showing you this? Because we're going to be looking at genes, specifically FH genes from patients, and genes are made up of DNA. So we see that DNA has base pairs. Okay, adenine base pairs with thymine, guanine with cytosine. So we see that with these base pairs, there's always a big base, okay, paired with a small base. And these are paired by hydrogen bonds. Remember, dashes usually are not covalent bonds, but hydrogen bonds, okay? And so we see an AT base pair has 2 hydrogen bonds, a GC base pair has 3 hydrogen bonds. And then these bases are attached to a sugar phosphate backbone, which is not really in detail in this picture, but we'll see that on future pictures, okay? But yeah, the base pairings bring the 2 sugar phosphate backbones together, but it's the base pairs that will determine if someone has the high cholesterol gene or not. This is looking at a single strand of DNA. Now it's more emphasizing the phosphate and sugar backbone that we did not see on the previous slide. So you have a phosphate, your deoxyribose sugar, phosphate, deoxyribose sugar, phosphate, deoxyribose sugar. And then they just minimize the bases as abbreviations here. And it's only showing you 1 strand. Okay, but it does emphasize the sugar phosphate backbone has phosphates with a negative charge, and that's going to be important later for separating DNA by cut DNA pieces by size to determine if someone has the mutated allele that dictates high cholesterol, or the normal allele that dictates regular cholesterol levels. So for FH gene analysis, they have found that the mutated FH gene, which is a dominant mutation, which overshadows the non-mutated version and gives people high cholesterol, can be cut by a restriction enzyme into 2 pieces. The normal FH gene, which does not cause high cholesterol, cannot be cut with the restriction enzyme and remains as 1 bigger piece. Okay, and so it's bigger than the 2 fragments from a mutated gene. However, those 2 fragments from the mutated FH gene, they should add up in size to the uncut DNA of the normal FH gene. So a little bit about restriction enzymes. However, I do provide a video for you to listen to and watch as well. So restriction enzymes are enzymes that cut DNA between specific base pairs. So they care if it's an A versus a T, an AT base pair or a GC base pair, and recognize specific sequences. So what comes before that AT base pair, it cares if it's a GC or another AT base pair. DNA is composed of nitrogenase bases, okay? And if we look back on a previous slide here, those nitrogenase bases can be adenine, thymine, guanine, or cytosine. And they contain sugar and phosphate. The phosphate of the DNA gives all DNA a negative charge. As a result, we can separate DNA fragments by size, which can be in base pairs or kilobase pairs, without charge interference. Restriction enzymes cut DNA at specific sites called restriction sites. Bacteria use restriction enzymes to protect themselves from inviting viruses. For example, there's an enzyme called BAMH1 that looks for this sequence GGATCC in DNA in cuts as shown. It likes to cut between two Gs, and then it gives a staggered cut. Now this ATG is base paired with the TAC here. I just couldn't show the hydrogen bonding there. Okay, and then this fragment here came from above. Okay, and it's still paired with base paired with this one, so it's not 4 pieces. It's 1 piece and then 2 pieces. And then we talk about pairs. Well, only 3 bases in this fragment is paired. So this is 3 base pairs, and then this, if you count this one, this is 11 base pairs. Now this does have a part that's single-stranded, and so that's called overhangs or sticky ends. Here I have some videos that explain about restrictions, enzymes, and sticky ends from YouTube, as well as the process of electrophoresis, which separates DNA by size, using the fact that all DNA has a negative charge. Gel electrophoresis, okay, which this is a little cartoon of. I'm showing you results you would obtain from the BAMH1 digest I showed you earlier, which is here, okay, where I said you get a 3 base pair band and 11 base pair band. Here's a well where you load your DNA, okay, and then one of the videos shows them actually pipetting into this well. You have your gel and electrophoresis unit hooked up to a power supply, and over time, the smaller fragments will move faster towards the anode, which has the opposite charge of the DNA. So, the anode has a positive charge to attract the negatively charged DNA phosphates, and larger pieces will not move as far. So you get separation of DNA by size. And then on here, I give you 2 more videos on DNA gel electrophoresis to determine paternity. It's kind of cute. It's probably too young for you, but she's looking at guppies. And to distinguish alleles. The second video is closer to what we're doing with the high cholesterol gene in people. So today we're going to analyze DNA from 3 patients in theory. One is FH recessive. They have the normal LDL receptor, has both copies of the normal gene, and it cannot be cut by the enzyme used, so it stays intact as one big piece. Another patient is FH homozygous dominant, so both copies are mutated, and both copies can be cut by the enzyme into 2 pieces. Okay, the sum of those 2 pieces should be similar size to the previous band for the FH recessive individual. The third patient is FH heterozygous, and only one of the genes will be cut into 2 pieces. The other one will remain intact. So therefore, you'll get 3 pieces. One piece will match the FH recessive individual. The other two pieces will be the same as the FH homozygous dominant individual. And so this person will have DNA fragments that match both of these. So the first patient will have one piece, the second patient will have two pieces, the third patient will have three pieces. What would these gel results look like? Okay, so if we were to draw this, here's our rectangle, it's a little crooked. Okay, so we can have the one individual whose DNA is not cut here. The next individual which gets two pieces could be here, and let's say here. And the other person which is a heterozygote, it would have one, like the individual that's homozygous for not having the mutated allele, and the other one is other gene that they inherited is the one that can be cut, so you get three pieces. So if this one over here on the far left was 100 base pairs, we can suppose this one here, this top one in the middle lane, was 60. And let's say this one is 40. We can see that no DNA was lost, but they have a site that they can be cut here, but not in this patient. Over here, this is the hybrid person or heterozygous person where they have the 100 base pair band for the one gene that can't be cut, which they inherited from one parent. The other parent gave them the one that can be cut by the DNA and encodes high cholesterol gene, and this would be 60 and 40. Okay, so that's how it would look. Hopefully yours won't look as crooked as mine. The goal is we are given purified FH genes from 3 patients. How did they be able to localize only these genes? This was done by a technique called polymerase chain reaction, but you're not responsible for this technique. But that's a very good technique to only copy one gene and study only one gene. Each patient's FH gene has already been cut by the restriction enzyme if it has that restriction site. We will separate the 3 patients' FH gene by size to identify which FH gene combinations each parent inherited using gel electrophoresis. So we already have a prediction here showing you that the one that has the 2 copies of the recessive gene with regular cholesterol will not be cut. The individual that has 2 copies of the high cholesterol gene will be cut. And the individual that has 1 copy of each will have 1 uncut gene, and the other gene is cut. This is just outlining how the gel would have been made. It's made much like Jell-O. You might remember Jell-O comes in a powder, you mix it with liquid, you either boil it on the stove or microwave it, and then you let it cool, and as it cools, it becomes solid. But you don't need to know the outline of making the small little Jell-O mold for gel electrophoresis since you aren't actually doing it in the lab. But you would have been loading your samples on your gel, and those videos I pointed out earlier would show you how to load your gel, which is the semi-solid like Jell-O. And so sometimes they have a molecular weight standard, which helps you determine what size your genes are. And then as controls, they put the normal FH gene, which cannot be cut, and the mutated gene that can be cut into 2 pieces, plus the 3 patients. And then one of those will mimic the normal FH gene and be cut into 2 pieces. The other patient will mimic the mutated FH gene where they have both copies mutated, so you get 2 pieces. Another patient will have 1 of each, so you would get 3 pieces. as I illustrated before. Here they're talking about once you apply your DNA samples, which I show a video of someone else doing by Biorad, you run it at a certain voltage and for a certain amount of time, and then you can either stain the DNA gel, electrophoresis gel, after it's been running. Sometimes people stain the DNA before they added it to the gel. This is an example of some sample results here. This would be a molecular weight standard where they know the sizes of the DNA here. It's nothing to do with the patients. And then we have DNA sample that's not cut. That would mean this person has both copies of the normal cholesterol gene. This is an individual who has 2 pieces. They have both copies of the bad gene, or both copies that says give them high cholesterol. Again, this one is this lane 1, 2. Lane 4 is the same as lane 2. This is an individual that has both copies of the normal cholesterol gene. Now look at this one. This is the first time we saw 3 bands here, drawn much better than my gel. So this one is a hybrid of this first individual. So it has 1 copy of the normal gene that gives you regular cholesterol levels or normal cholesterol levels less than 200. And then these 2 bands represent the mutated copy that gives high cholesterol. This final DNA sample is the same as the 3rd DNA sample, where this patient has both copies of the high cholesterol gene, and both copies were cut. So, are you going to inherit the FH disease or not? This disease is dominant. You can have high cholesterol disease if you're heterozygous, where you have 1 copy of the FH gene and 1 copy of the FH gene. Now notice I changed the case of the letters. Capital F, capital H means dominant. Little f, little h means recessive. Or you can inherit 1 copy of the dominant gene, big F, big H, 1 copy of the recessive gene, little f, little h, which does not give you high cholesterol. You can inherit both copies of FH, which would be the dominant disease form. To not get high cholesterol at all, well, from your genes that is, you would need both copies of the little f, little h to have the recessive condition. Now, but that doesn't mean you can't get high cholesterol from what you eat. So here we're going to link genetics to those high and low cholesterol and look at the chances of inheriting the high cholesterol gene or high cholesterol. So here we have 1 parent is homozygous for high cholesterol, as shown by big FH, and the other is homozygous for low cholesterol, little FH, little FH. And this is the recessive condition. The only way you can get the recessive condition is if you have 2 copies. So what are the chances of them getting high cholesterol? So you fill in these squares as shown here. In this case, anything to the left of the squares, you go from left to right. Anything above, go top to bottom to fill it in. So this first square would be big FH, little FH. So they're heterozygous and would have high cholesterol. This individual would be big FH, little FH. So they would also have high cholesterol but be heterozygous. This individual will be the same, big FH, little FH, high cholesterol but heterozygous. This individual is high cholesterol as well, and it's also heterozygous. So in this case, they're all 100% high cholesterol because they have at least 1 FH, big FH. They have at least 1 big FH, so it's 4 to 0 is the chance of high cholesterol to low cholesterol, or 100% high cholesterol to 0% normal cholesterol. Here, we're looking at if 1 parent is heterozygous, as shown on the top, but they still have high cholesterol, And one's homozygous for normal cholesterol, little FH, what are the chances now of the children getting high cholesterol? Well, this square is big FH, little FH, high cholesterol. This individual on the top right is low cholesterol, well, not low, normal cholesterol, because it has little FH, so normal cholesterol. This individual is big FH, little FH, so high cholesterol, but heterozygous, like the top one above it. And this one is the same as the square above it. It's homozygous, recessive, or has normal cholesterol. So in this case, it's 50% to have high cholesterol, but they're heterozygotes, and 50% to have normal cholesterol. Now we can also look at it. Here's another cross where we have a heterozygous individual, but they're still high cholesterol because big FH is dominant over little FH. And the same here, this other parent also has high cholesterol, but it's heterozygous. Now what are the chances of having high cholesterol? We have big FH to big FH, whoops. High cholesterol, homozygous and high cholesterol. This one's heterozygous and high cholesterol. This one's heterozygous and high cholesterol. This one is homozygous recessive and low cholesterol. I keep saying low, I'm sorry, it's normal cholesterol. So 1, 2, 3 squares are high cholesterol. It doesn't matter that 2 of the squares have a little FH. As long as they have a big FH, it's high cholesterol. So you have high cholesterol 3 to 1. So these 3 squares have high cholesterol and this has normal cholesterol. So it's a ratio of 3 to 1 or 75% to 25%. So 75% chance of having high cholesterol and 25% of having normal cholesterol. Okay, so that's how you do the Punnett squares. You will have to do at least one per the lab manual to answer one of the questions in the lab manual. If you have any questions, of course, please email me or Zoom for this laboratory report. And I hope you enjoy the little videos I put in here. Have a good day.